forward this up to the cloud. Hopefully I'll be able to get it back down later. And I'm going to go to the whiteboard for this. Okay, so today we need to start to talk about T cell activation. So remember last time when we had kind of had a picture there of, right, so the immune response, and it be divided into those two parts, B cell response and T cell response, right? So today we're going to focus on the T cell response. Now, the T cell response is a lot more complicated than the B cell response. So we're going to start same old, and we're going to talk about T cells are derived. They're called T cells because they go to the thymus. So their stem cell is still in the bone marrow and the fetal liver. But after they come out of the bone marrow and the fetal liver, and they've started to differentiate into either what we would call a CD4 positive cell or a CD8 positive cell, those cells are going to be destined to become T cells. So I'm going to put CD8 as a lollipop here with a little round top, and I'm going to put CD4 as one with a triangle. Okay, so this is a CD4 positive cell, and this is a CD8 positive cell. And try to remember, guys, you know, we're giving you, this is a survey introduction to immunology. This is by no means completely extensive, okay? In the real world, there are also cells that are both CD4, CD8 positive that come out of the bone marrow. There are cells that are both CD4 negative and CD8 negative and come out of the bone marrow as well and are destined to become T cells. We're not going to discuss all of those, all the minutiae. We just don't have time. So similar to our B cells that we said when they come out of the bone marrow or the fetal liver, they're essentially pre-B cells. So are these guys. These are like, they will become T cells of some sort, but of which type, it's yet to be determined. They go to the thymus, and in the thymus, we say they get educated. And what that really means is they acquire their T cell receptor, and they're sort of put through a check system so that we don't want any T cells to leave the thymus that are specific for self proteins. We only want T cells to leave the thymus that have a receptor, sort of like antibody is a receptor on those pre-B cells. These T cells would have a receptor for antigen plus either class 1 or class 2 MHC. So they can't leave until they've been screened. And then only the ones that are determined that they are not going to bind to self, those are the only ones that leave the thymus and then go out to the lymph. Okay. Sorry, it's a little bit noisy because I'm out on the porch. There's, of course, today the township is coming around to pick up leaves and brush. Now, before we can talk any more about T cell activation, we have to backtrack a little bit and we have to talk about the major histocompatibility complex. So we abbreviate it capital M, capital H, capital C. Histocompatibility complex. In humans, so this was discovered in mice, and there's 3 categories to it. We call them class 1 MHC with a Roman numeral like this, class 2 MHC, and class 3. We're not even going to talk about class 3. Okay, we're just going to focus on class 1 and class 2. Now, as I mentioned, these were elucidated or worked out in mice. In humans, we usually refer to them as the HLA. So if you've heard that before, if you've heard it referred to as the human leukocyte antigens, that's what that HLA stands for. Okay. So like I said, we're going to focus on class 1 and class 2 MHC. Class 1 MHC, this is unique to each person, each individual person. And this protein is on the surface, so it's in the cell membrane of all your nucleated cells. This is what tells your body, basically, this is self. Okay, this is not foreign antigen. This is not non-self. This is self. And certainly the people that you're related to, your parents, your siblings, right? You're going to share more of the protein in common with them, but it's unique to each person unless you're an identical twin. Okay, and even then there are some minor differences. The class 2 MHC, this is a signal, basically. This is a protein that's just on the surface of lymphocytes. Okay, so you will see this on the surface of B cells, T cells, macrophages, dendritic cells, right? And they look different, the class 1 and the class 2 MHC. And your textbook draws them kind of like this. So for the class 1 MHC, I'll give us a little phospholipid bilayer here. Can you guys turn your microphones off, please? I'm getting a lot of background noise. Thanks. Your audio. So here's the cell membrane, right? And then in your book, they show class 1 MHC sticking out there, and they've got it drawn, I believe, on on page 414. They've got it drawn as like 4 blue circles, and then this little yellow piece. Sorry, I guess it's only 3. And then this 4th one sitting here like that, right? So that's kind of what class 1 MHC looks like. And again, remember, that's on the surface of all of your cells. The class 2 MHC looks very different. Class 2 MHC, if I give us another cell membrane here, right, phospholipid bilayer, and I would put, these tails are what holds the protein in the cell membrane, right? And then the class 1 MHC, the class 2 MHC, they kind of look like this. They show it kind of like, oops, I'll go back to a pen, like this. And they put a little gap in there because that's where it's going to bind antigen. Okay. And they actually make it as 2 separate proteins. Looks like kind of like that. So I would say that's pretty good representation of class 2 MHC. Now, B cells, of course, are self, so they're going to have class 1 on them. So are T cells, right? So they're going to have them on the class 1 MHC on the surface as well. This is the tricky part to remember, is this class 1 is on all of your cells, all your nucleated cells. This is why transplant is so difficult from one person to another, because your class 1 MHC is yours and it's different from everybody else's. And so your body would recognize that as a foreign antigen and make an immune response to it if you get a kidney from someone else, say, or a liver or a lung. Okay, now there are more things on the surface of T cells. I'm trying to get another whiteboard here. I could add one. There we go. So if this is a T cell here, and this T cell is destined to be, that's the nucleus, this T cell is destined to be a cytotoxic T cell, which I'm going to label as T for T cell, and like a subscript of a C for cytotoxic T cell. In addition to having class 1 MHC on its surface, because it's a cell of the immune system, so I'll draw those 3 circles. That cytotoxic T cells usually also have, they have a T cell receptor. So I'll draw that kind of like this. And that T cell receptor is going to have, you can think of it as sort of having 2 parts. It has a place or a part where it's going to be specific to bind to a particular antigen. And it also has a part that is going to stick out. In the case of a cytotoxic T cell, it's also going to have CD8 on its surface. So I'm going to draw CD8 again, like kind of like a lollipop. okay? In addition, every T cell near their T cell receptor also has this other group of molecules, CD3, and those are molecules that mark early T cells. So I forgot to mention what the CD stands for. It stands for clusters of differentiation, and your book mentions there's hundreds of them, and they're markers, they're proteins that cellular biologists use to figure out what cells have the potential to become what other cells for differentiation of cells, both during development and in adults. Okay, so all T cells are going to have a T cell receptor and CD3 on their surface, and class 1. And if you're a cytotoxic T cell, you're also going to have this CD8 cluster differentiation 8 on your surface most of the time. I wanted to talk about cytotoxic T cell activation first because I find it a little bit easier to understand and process. So like I said, we're going to talk about that one first. I'll call it activation. Right. So here's our cytotoxic T cell with all its stuff on its surface. And the cell that is going to trigger a reaction in this cytotoxic T cell, we call that cell a target cell. So I'm going to try to make it look a little bit different in shape. And the target cell is part of the host. So there's the nucleus with a nucleolus. And let's just say it's easy for me to think of it, let's just call it a liver cell, okay? And let's say that this cell has been infected with a virus. Let's say it's hepatitis. So we'll draw a virus out here on the surface and on the other side, and I'm going to put little red circles on the outside of this virus. Okay, so there's the hepatitis, human hepatitis virus. Virus goes in, just like we talked about in chapter 6, take off its coat, and then what happens inside our host cell is that host cell is on its surface. It is going to put in next to some class 1 MHC, it's going to put some of the virus particles like that. It's going to join them together and put them on the surface. And that is going to be a recognition signal for our cytotoxic T cell. I'll draw it again over here because we can make it look more like it's going to fit into the receptor. So the antigen portion, whatever piece of this protein from the hepatitis virus that the target cell ingested and re-expressed on the surface, that's going to bind to the T cell receptor, okay? And the class 1 MHC is going to serve as recognition. That's the CD8 is going to bind to that class 1 MHC. So it's a two-part binding. We have to bind antigen, we say, in the context of self, and the self in this case is class 1 MHC. When this occurs, if this occurs, okay, the cytotoxic T cells, they're called that because they kill, they lyse and destroy other cells. How do they do that? Well, inside of them, they have a compartment that is filled with, and I'll just draw it with little dark dots here. And it's filled with enzymes, and these enzymes are called perforins because they perforate. It means they make holes in the target cell. And granzymes because they are enzymes that are granular, right? They look like dots inside the cytotoxic T cell. So I should say when or if, right? When the cytotoxic T cell receptor binds antigen plus class 1 MHC on the target cell, or you might even want to change that when to an if the cytotoxic T cell receptor binds antigen and class 1 MHC on the target cell, then that cytotoxic T cell releases those granzymes, and perforins. And what those are going to do is, I'll make that big and in red, they are going to lyse and destroy to destroy the target cell. That is tissue damage. So that is going to then stimulate inflammation. That's going to cause more phagocytes to come to the area. Those of you that already know this process, right? The phagocytes are going to become antigen presenting cells for other T cells, both cytotoxic and potentially helper cells. Questions about that before we go on? Yes, no?
No.
Yeah. Quick question: when you you just erased it, but on the host cell, you had the red. You had the red coming out to the left.
Right. That's this guy. He's supposed to be a piece of the antigen that has gotten processed from the virus and put on the surface with class 1 MHC for a recognition signal for our cytotoxic T cell.
Okay, so he was kind of booted out.
He's attached to that class 1 MHC.
Okay.
The target cell very specifically binds pieces of the virus protein to class 1 MHC and literally puts them out on the surface to say, hey, everybody, I'm self, but I've been infected with something that's non-self, and so I need to be destroyed.
So kind of like waving a red flag?
Kind of like waving a red flag. Yep. Come over here and obliterate this because I'm going to become more of a problem for the whole host if I stay infected and the virus multiplies.
Got it.
Right?
So that's what they were talking about in the book, kind of like a street sign or a stop sign, that like a little flag that waves, and then the TC cell can come and pick it up.
Yes.
Okay.
And I've drawn this in a very simplistic manner. This target cell in your textbook, they don't even show it as a target cell, they show it as a macrophage, right? Because sometimes it will be a macrophage. But let's keep it simple because we're doing the basics here. All right, the helper cell, that one is a little more difficult, or there's a little more finesse that involves when you stimulate a helper cell. And the reason is because the sort of premature helper cells, or the CD4 positive helper cells, when they get triggered, like a B cell, they can be sort of clonally selected, and they can then differentiate into multiple types of other helper cells. Now, H for help in their name, right? They are not going to release any chemicals to kill any other cells. All the things that they're going to release, all the interleukins, all the cytokines they release, their job is to help booster the immune response. Okay? So here's our helper cell. Draw him. Again, it would have a T cell receptor on the surface. Right? So I'm going to draw it like that. And I'll keep my antigen a circle because that's easy for me to draw. And it's going to have some CD3 molecules next to that T cell receptor. I'll remind us here in green that this is the T cell receptor, the TCR. And then T helper cells are going to have not CD8 associated with them, but they are going to have cluster of differentiation protein number 4 associated with those T cell receptors. So I'm going to draw that black here, and I think before I made a triangle on the top of CD4, so I'll try to keep that the same. Okay. And again, this is more complex than I'm showing you, and if you look at the picture, especially the close-up of the picture from on figure 1519, you'll see there are other accessory molecules that have to bind together in order to trigger this T helper cell response. But I'll just label that one as CD4. Would it have MHC1 on its surface? Absolutely, it's a cell that belongs to you. So I'm going to draw one of those over here, right? Would it have class 2 on its surface? Yes, because it's a cell of the immune system, and that's partly how they talk to each other. So here's our, if you want to think of it like even as a precursor helper cell. I'm looking for the figure. The sort of summary figure is 1511 in your book, and this would be the top panel. The part that I drew just for the cytotoxic T cells, kind of the bottom panel. In order to trigger a helper cell, you need to have a macrophage or some sort of what we call an antigen presenting cell or antigen processing cell. The abbreviation is APC, right? And I'm trying to draw this one to look like a macrophage, right? And what this macrophage does is it takes the antigen, so we'll make it a bacteria, that's easy, and I'm gonna put, again, circle antigens on its surface because I can only draw a circle, triangle, square, right? And so these macrophages, of course, are gonna be involved in phagocytosis. They're gonna ingest our bacteria here, and they're going to put it in a phagosome, and then it's going to fuse with the lysosome, right? We know this story from the last chapter. And what is going to happen is pieces of this antigen, again, are going to be literally joined to some class 2 MHC, which are supposed to be these things here, and again, put out on the surface of the cell. So I'm going to try to put the class 2 MHC over here and make it look like it would bind to triangle. And then I'm going to put, attached to that is going to be our bit of antigen that's going to fit into the T cell receptor. Okay. So again, it's a co-interaction. To stimulate this helper cell, it has to bind, this T cell receptor has to bind to antigen in the context of class 2 MHC, which is what that's supposed to be. Class 2 MHC, right? And then this CD4 molecule is going to grab on to the class 2 MHC to kind of seal the deal. Now, this cell does not release any perforins and granzymes. What this cell releases are cytokines, as well as the macrophage. When it binds to a T cell receptor, it also is going to release some chemicals, particularly IL-2, which stands for interleukin-2, IL-4 for interleukin-4. And those, the receptors for those over here on our T cell, and I'll make them squares, the interleukins. So the macrophage is going to release those. They're going to come over here and bind to these, I'll just put interleukin receptors, R for receptors. And that's what's going to trigger this T cell then to differentiate into potentially, so actually I should write some words here, right? So if a T helper cell, TCR, binds to antigen plus class 2 MHC, then interleukins are released from the macrophage that are going to stimulate, let's say, clonal expansion and differentiation of our T helper cell. And all of the different kinds of cells it can become would be a T memory cell for the next time. It can become a, I'm just going to put up full-blown adult T helper 1 or T helper 2. It is not important to me that you know the differences between the cytokines that they produce. The T helper 2s specifically, though, they are necessary. They help stimulate B cells. There are also these cells we call T17 helper cells. They are involved in inflammation. And then the last ones we call Treg or T regulatory T cells. We used to call them suppressor T cells back in the day, like in the 90s. We don't call them that anymore. Their job is to regulate the immune system or help tamp down the response when it is over, right? So I'm going to kind of just draw these all as different kinds of cells. And they do not like directly they don't directly produce antibody. They don't directly produce chemicals that are going to kill anybody, right? Their job is to help, and they help by all of them secreting different interleukins that are going to either increase this response, in this case, in this case, or in the case of a T regulatory cell, of course, they're going to tamp down the response. Okay, so IL stands for interleukin. Those are special cytokines that just talk between the leukocytes. And I think we're up to like IL-20. Okay, so we're not going to go through all of them and all their functions. It's not important for us. What's important is how this sort of early T helper cell gets turned on, and then what happens to it once it actually binds antigen in the context of self, antigen in the context of class 2 MHC, and that is presented by an antigen presenting cell, or some textbooks will call them antigen processing cells. It means the same thing. All right, questions about that before I go on? Yes, no, maybe so. I know it's a lot, but you can do it step by step, right? One part of it at a time. So what's on the surface, what it binds to, how did those things get on the surface, what happens if they bind, right? What becomes of this cell, and what can it do for the immune response? These T memory cells are going to be super important. They're important for the next time you see this particular infection. All right, I'll let you chew on that a little bit. And I'm going to add another whiteboard and talk a little bit about, I put on there IL-10. I can't draw that picture for the primary and the secondary response. You should look in your book for that. It's primarily discussed as an antibody response, an immunoglobulin response, and I can draw a rough approximation of the primary response. Looks like this, second, and then there's a break in exposure, and then the secondary response looks like that. And I can probably rough approximate the immunoglobulins here if I do it while I'm looking at the picture. So in the primary response, which is here, the first response to the antigen, you're going to see a very small level of IgG, but in the secondary response, sorry, this is the IgM. The IgM comes first and is a low amount. The IgG comes later and is a higher amount. Okay. In the secondary response, the IgM is a little bit elevated and sooner, but it's the IgG that you get a spike of it much sooner. So the blue is the IgG, immunoglobulin G. And remember, IgG is the super immunoglobulin, or the poster child. It could do so many of those functions that we talked about in terms of what can immunoglobulin do for you. Neutralize, opsonize, present to macrophages. This primary response, this lag time here, I would say it's normally about 7 to 10 days. The secondary response, you get a really 3 to 5. So you get a much stronger response, much quicker the second time. The whole purpose of us even looking at this is to help us understand why or what the benefits of getting a particular infection are as far as protecting you from seeing that infection again. And basically this is the point of vaccination, is to cause an artificial primary response that will help you make a really strong secondary response if you see that antigen again. Okay. Which takes us to the different types of immunity. I don't care for the picture in your book. It has way too many words on it, in my opinion. So, right, we sort of divide up immunity into acquired and innate. Innate immunity would be things that you don't have the genetics to make a response to. So for example, you're not going to get kennel cough because you're not a dog, right? So that would be what we would call, what an immunologist would really refer to as innate immunity. Acquired immunity is immunity that you have that you acquire during the course of your lifetime because of different antigens that you've been exposed to. And it can be divided into naturally acquired immunity or artificial. And then each of these can be divided into passive and active. And again, there's a table that shows this in your book. It's table 15.4. Naturally acquired passive immunity only occurs when you're a baby, when you're a fetus, or when you're a newborn. Really? And because it's passive because these are immunoglobulins that you're going to receive from your mom, right? Either through the placenta if you're a fetus, or in breast milk if you're a neonate. Naturally acquired active immunity, this is when you get an infection, right? You are exposed to COVID-19, you cut yourself and are exposed to a bacteria, you get a cold from someone in your family, whatever it is, right? Naturally acquired active. You are going to produce B cells that are going to turn into plasma cells that are going to produce lots of immunoglobulin for you, and you're going to make T memory cells for the next time you get this infection. The second time you are exposed to it, most of the time you don't even know you're sick because your body makes such a strong response. Artificially acquired passive. This is also like naturally acquired passive, is a real rare or very limited incidence. This is when you passively, so like via IV, you would receive immunoglobulin from people who have already recovered from this disease. This is difficult to produce because it has to be extensively filtered, and it works best when the immunoglobulin is specific for the particular disease that you have. And most of the time, that is difficult to do. Okay, very limited. Artificially acquired active, that's a vaccination. Okay, so artificial, because you didn't fall down and scrape your knee and get exposed to tetanus. Someone injected tetanus proteins or tetanus toxoids into your system on purpose so you could make an active response, right? And again, because it's an active response, you're going to produce immunoglobulin, you're going to make memory B cells, you're going to make T cells, maybe cytotoxic, maybe memory. which brings us to the last part of the chapter, which is about vaccines. Right. And basically, we divide vaccines into two categories, what we call whole cell or whole virus. And this isn't complicated. It's just like it sounds. It's the whole organism. Clearly, it would not be wise to give people a large dose of live organism because it might make them sick and in some cases might overwhelm their immune system. So these vaccines are either going to be, we say, killed, or if it's a virus, we don't say killed, we say inactivated. And that is accomplished by usually by heat or by chemicals. Or they might be what we refer to as live attenuated, which means they have the ability for some limited multiplication, but they're usually maimed so much that they can't cause a full-blown infection. Okay. The advantage of the whole cell or the whole virus is that you get all the antigens, right, from that organism. The disadvantage is that if you didn't kill all of them, or if you didn't inactivate enough of them, right, the main disadvantage is the potential for an actual infection, right? So this one we always consider it has more risk. Now, so what people decided was like, oh, well, if that's too dangerous for the overall population, meaning maybe 1 in 500,000 doses, someone's actually going to get sick, or 1 in a million doses, if that's too much risk for us to live with as a population or a society, a better option might be to produce what we call a subunit vaccine. And again, that's just like it sounds. It is only part of the organism, either the bacteria or virus. It is usually the proteins that we have identified cause the best immunoglobulin response and the best T-cell response. Now, the difficulty is, how do we know which ones cause the best response, right? If we're using an animal model, maybe the mice respond to different antigens than we do. If we're actually using a human model, different populations of people are going to respond differently, meaning different ethnicities of people. And if you don't test them all, then you don't know all the potential antigens. So the subunit vaccine usually is produced, and then it's usually improved and improved and improved and improved over time. And I listed out some of the ways that you can get these subunits. You can most simply, of course, you can grow the organism, and then you can, lyse it or break it up and purify the proteins. What's more common now is, of course, they are what we call like semi-synthetic, in that they are some of the proteins that have been modified in the laboratory, or you can actually genetically engineer, right, a cell like a bacteria to only produce some of these particular proteins, and then it's easier to lyse them and purify them. The downside of the subunit vaccine is it is only some of the antigens. You are usually going to require multiple doses to get a strong response over time. And if people don't get all the doses, think about the childhood vaccines, right? If you don't get all the doses, then you're actually not as protected as you might think you are. And then I put last thing on your on the list there on the handout was I made a mention about herd immunity, because that's the goal of any vaccination program, is to not immunize every single person, because there will always be some people where the risk is too high, but to immunize enough of the herd, right? Enough of the population that the organism doesn't have a reservoir or very limited reservoirs, and then of course has very limited ability to transmit to an unvaccinated individual. So this is the goal, is to produce this herd immunity. And we've seen what's happened with, for example, the measles vaccine, particularly in areas where people have decided it's too dangerous to immunize their children against the measles because they falsely believe it has something to do with autism, that the incidence of measles in those populations has skyrocketed. And measles is actually a rather infectious disease, and it can kill you. And when it is, think about it, when it's in the reservoir, it has opportunity not only to be transmitted, but it's a virus. So it's going to mutate and has the potential to become a more virulent version, or a version the rest of us aren't actually immunized against. So again, this is the goal, right? All right. I'm going to stop the sharing and ask if anybody has any questions.